Hi folks, welcome back to Backyard Microscope. Today we have a very exciting episode this is episode number 5, and this topic is near and dear to me because it's what I did my master's thesis on, and this took me 2 years to complete this project. Today we're talking about trematodes. They're tiny little microscopic worms that live in aquatic systems, and it may not be in your backyard, but I'm real close to the actual estuary where I caught the animals that are part of this life cycle of this crazy microscopic worm. Now, as I mentioned, I published my master's thesis on this. I also published several other publications on different discoveries. In fact, I got 2 journal covers from the Southern California Academy of Sciences with papers published on this very interesting organism. I also published in a journal called Parasitology Research, which is from Germany, and got a really neat article published in that. And then, of course, another color journal cover, which I'm very proud of. So I was able to discover many things in this organism, and there's still a lot of work to do. Now, beyond the fact that these are very cool organisms and that they're microscopic, they have a crazy life cycle, which we're going to talk about. Our life cycle is very simple. We grow up from a baby with parents, and we don't go through other organisms to complete our life cycle. But these guys go through 4 and sometimes 5 different organisms as hosts to complete their life cycle. Now, one other really interesting fact about these microscopic worms, these are called trematodes, or maybe you've heard the term fluke, is that they generally do not affect the health of many of the hosts that they're in. And so, in fact, with the fish that they infect, you can have them in the brain, you can have them in the heart, in the stomach, in the mesentery, in the gills, in the liver, in the fins. And some folks have called these fish that are parasitized by these different trematodes floating zoos, because they're just little floating zoos full of these different species. And one of the things that's really interesting about this trematode is that depending on the species, it will generally only infect a specific fish, and in that fish a specific organ. For example, Ascocotyle pachycystis, I know, is always going to be in the heart. It's always in the heart of specific fish, and others specifically infect the brain or the liver or the gill. So very interesting that the parasite is tied to the actual organ. So we're going to talk about the life cycle in specific. You'll see a diagram of the life cycle. Another thing that's very interesting is that this guy goes through a snail before it goes to the fish, and it can castrate the snail. And the question is, how does it do that, and why? Why does it castrate the snail when it's in that host? They also make a cyst. They make a little arc that they're inside of, a little walled ball of material that's very interesting. Some are very thick-walled, some are thin-walled, and the development of the worm is arrested. It's actually stopped when it's inside the cyst, because it's waiting for the rest of the life cycle to continue, because the fish has to be consumed by a warm-blooded organism. A bird, typically these are found in birds, but also rats and other warm-blooded vertebrates can eat these fish raw, and they become infected also. One interesting thing about the cyst that you're going to see images of is that the cyst is hydrochloric acid resistant. And that means when the bird or the muskrat or the rat eats these fish raw, it passes right through the stomach where hydrochloric acid is present, and so it's resistant to that, so it passes right through the stomach. The other thing that's very interesting about this organism is that it's an immune stealth organism. It doesn't really affect the immune system of the host that it's in. So the immune system barely even reacts to the presence of these trematodes inside those organisms. So I hope you enjoyed this episode. We've got some neat video to show you and some neat photographs, and let's get started. Here is a diagram representing the life cycle of this very interesting trematode worm. For a second, just forget about the human diagram here, and we'll concentrate on the red lines. But if you look down at the bottom of the diagram, there's a yellow number 1, and that's where the eggs come out of the final host, which we call the definitive host, and land on the mud flat floor. And they may go through a few changes before they get ingested by number 2 to the left, which is the snail, Cerathidia in my case, and they pick up these eggs, and the eggs develop inside the snail. Interestingly, the parasite often castrates the snail, which is a subject of great interest in the literature. Nevertheless, it develops and it comes out of the snail to number 3 you see on the left there, which is the swimming stage. This is a larval swimming stage called a cercaria, and it swims in the water column until it gets caught up in the respiratory current of the fish that it's going to infect. And remember, it only infects specific fish. So it goes through the mouth in the respiratory current, it finds the gill, it attaches to the gill, it drops its tail, and then it burrows into the gill until it finds the blood system. And once it finds the blood system, it tumbles through the blood system until it arrives at the exact organ that it's supposed to be in the fish. It is there that it makes a cyst that protects it, and the development is arrested until it gets eaten by the next host in the life cycle. Now, there are 2 important things at the top of the diagram I need you to pay attention to. Species/organ specific. These trematodes only infect specific fish and a specific organ within that fish. Number 2, to the right of that, you see HCl resistant. That little cyst is resistant to hydrochloric acid so that it can successfully pass the stomach of the next host. Now here's a photo of Cerathidia californica, which is the typical snail that's found on the mudflat floors in these estuaries in California. Now it is in that snail that this structure is formed, sort of a mother brood sac. It's called a rhedia, and it produces all those swimming larvae that come out of the snail and swim in the water column up to the fish. Here's an actual picture of a cross-section of the rhedia, and you can see all those tiny structures inside, which are the developing cercaria. Here's a scanning electron microscope picture of that cercaria, that swimming larval stage. You see a very strong muscular tail on one end and a very strong oral sucker on the other. Here are some of the fish that are often infected with these trematodes, this one being the sailfin molly, and there's other estuary-like fish where these short bait fish are often parasitized. Another is called Poecilia, and one that looks like this and behaves in a similar way is Gambusia, the mosquito fish. So another fish that's infected with these trematodes. And finally, one that I worked with a lot, the killifish. The California killifish was one that I worked with a lot. In fact, I found Texas killifish in South Padre Island, Texas that had brain parasites, and that's one that I'm working on now. Okay, so let's get to an actual infection. And here you see the heart of the Poecilia fish. This one was actually collected in Ocean Springs, Mississippi, and you can see the bulbus arteriosus, which is this bag. I'm ripping away the rest of the heart now, but this bag is filled like a bag full of marbles of these trematodes, Pachycystis, Ascocatale pachycystis, which is a thick-walled cyst. Here's a little bit higher magnification, and you can see that this heart is just jam-packed with these trematodes. Now, these are living specimens, and you're going to see some of these. I'll isolate some of these individual cysts in a minute. You can see how thick they are, and note the wall. Now, here's a different variety found in a gill. You see a larger worm on the left, wrapped around itself. You can see it moving, and then on the right in a second you'll see these smaller trematodes. So these are alive. They're living inside the gill, and they're waiting to be freed from their little encystment by the conditions found in the vertebrate gut. It's looking for a temperature, a certain temperature. It's looking for a pH of about 7.2, and there have to be bile salts and trypsins in order to digest these things. And you'll also note here as we move, you'll see that they're between 2 blood vessels. There's a blood vessel on the right and one on the left, and here's a dark field shot, and you can see these moving a little more clearly. So their development is now arrested. They will not continue in their life cycle until this fish has been consumed by a worm, a vertebrate, a warm vertebrate that can support them in the intestine, and that's where they will mature. Now, when they mature in the intestine, they will make eggs and sperm, because these are hermaphroditic, and I'm just working on this bag to get some of these individual trematodes out of here so you can see them. And I'll magnify this one in a minute so you can see the individual worm inside. But note how thick the cyst wall is on this particular organism. So we know right away that this is Pachycystis when we see this very thick cyst. But you can see how difficult it is to get all of these trematodes out of here, and yet they will all survive. Now there's other areas that they infect the fish too, and for example, in the liver, and here's a thin section of liver from the California killifish. You see one trematode in there indicated by the black arrow. The green arrows show the very thin cyst wall, and the white arrows show the liver tissue. And notice that there's no inflammation as a result of this trematode inside the liver. The immune system doesn't even react. Here's another example of liver tissue. The white arrows point to the liver tissue. Green arrows point to the little bit thicker now cyst wall of a different organism, and the black arrow points to the trematode. So these set up shop very well inside of liver, inside of the stomach, inside of the mesentery, in the gills, in the heart, in the brain, and so these are found throughout the estuary. Here's now a gill section. The black arrow points to the parasite. The green arrows point to the thin cyst wall, and the red arrows point to the gill. And so these things set up shop, and they wait until the fish is eaten by the next host in the life cycle. Now, I also did some thin sections of Pachycystis, and you see those here. And these are stained with a very interesting stain called PAS, periodic acid shifts. And this actually shows the different layers in the cyst wall, and I did some electron microscopy of those layers, and you'll see those in a little bit. But these fish are barely affected by heavy infestations, even in the heart. Here's another trematode that is also in the heart. The green arrows point to the thin cyst wall, the red arrows point to the actual trematode, and so these pack these hearts like a bag full of marbles, and the fish is no worse for the wear. They perform very well, even though they're heavily parasitized by these organisms. Okay, I promised you some high magnification video of these things turning in their cysts, and here it is, ascocotyle Pachycystis behind this very thick cyst wall, turning on itself, waiting patiently for this part of its life cycle to be collected by a warm-blooded vertebrate that will move it along in the life cycle. As I mentioned to you previously, I did some transmission electron microscopy of the cross-section of the cyst wall of Pachycystis, and here you see several different layers. You see the host H on the upper left, and then you see OML, the outer marginal layer, and then three other layers, UM, ML, and TML, which I identified as separate, distinct layers in this cyst wall, and then MT, of course, corresponding to the metastracaria, which is the worm encysted inside this cyst. And so this was published in the German journal Parasitology Research. Now, here I'm going to show you more of these Pachycystis worms encysted, and I'm going to try to take one out mechanically. Now, normally what you would do is you would use a solution of bile salts, trypsins, and a pH of 7.2 to get these trematodes to come out under chemical excistation, or using a chemical to get them to come out or exist. But here I'm using a very tiny pin, a needle, with a fine forceps to try to mechanically separate these out. And as you can see, it's rather difficult to hang on to these. They're very slippery, but if you run this needle across them, eventually you break it open. And here I've broken the cyst open. However, I've kind of mangled the parasite within, and you'll see this parasite, but I kind of hurt him. I put a little cut inside of him, and so he's not going to respond very well. So we'll come back and do this again. One thing I did want to point out is that all of these trematodes in this family have a row of spines around the oral sucker, and this one has one row of evident spines. Sometimes you have to fix them and prepare them a little more to see both rows of spines. But this is the oral sucker, a very strong sucker with an oral row of spines. And now we finally come to what we call the definitive host, or the final host in the life cycle of this tiny worm. This is a heron, an egret, or other bird that eats these fish whole. This happens to be an egret, and it is in this bird that the fish is dissolved by the stomach as normal, and the cyst, however, being HCl resistant, passes through the stomach and travels to the intestine where the worm matures. And in maturing, these things grow a little larger, they develop a little more, they also develop internal organs. They are hermaphroditic creatures, and so they make both testes and ovaries. And because these mature worms have both testes and ovaries, they manufacture eggs and sperm, and so they self-fertilize. And here you see a photograph of a mature worm. You can see the eggs on the right side, the ovaries and the testes also. The red arrow is pointing to the oral sucker, and the yellow arrow is pointing to a very strong and muscular ventral or tummy sucker. And so because these are self-fertilizing, they can set up shop in the intestine, and they very happily pump out thousands and even hundreds of thousands of eggs a day into the marsh. And then the life cycle begins all over again. The egg is picked up by the snail, and off we go again with the life cycle. So very interesting creatures. Now here I was able to successfully exist one of these worms mechanically, and you can see how muscular they are. He's laying on his back, and what he wants to do is flip over to his tummy so that he can use both his suckers to move along and look for a place to set up shop in the intestine. Under the light microscope at about 500x, it's a little difficult to see that there are two rows of spines. So sometimes light microscopy is a good start, but then we have to go and use electron microscopy. Here's an electron micrograph of another species of ascocotyle, and these arrows point out clearly the two independent rows of spines adorned around the oral sucker of this organism. So some come with one row of spines, some come with two, some have incomplete rows, but generally they have spines around the oral sucker. There are many varieties to this group. Here's another one, and it's an adult. You can see the arrows pointing to the eggs, and these eggs will pump out into the feces of the definitive host, the bird or the raccoon, and that will go out hopefully into the mud-flat floor. Another species was discovered in Colombia, which I got to name after my professor, Dr. George Howe. So this is Ascocotyle howei with two rows of spines. But the electron microscopy is a lot of fun. Here you see a strong ventral sucker, tummy sucker, and a strong oral sucker with two rows of spine. And I was also able to discover Ascocotyle fagicula diminuta, which is this guy. The only other record for this was from a rat which was in a landfill in upper state New York. But here's a new species that has yet to be discovered, found at the Newport Back Bay in Newport Beach, California. And so I was able to get several publications out of this. Of course, 4 publications, 3 in the Southern California Academy of Sciences. And here I got a cover of one of the worm's oral suckers. And here is another cover showing the swimming cercaria. So this is a lot of fun, and there's a lot of work to be done. So come on in and join us. Okay, folks, I hope you liked our short introduction to trematodes, flukes, these parasites. I find them very, very interesting, but I'm left with a lot of questions, even in my studies, and maybe you could jump in and help to answer some of these questions. For example, why does it have such a complex life cycle? I mean, it's dependent on 4 and sometimes 5 hosts to get to maturity, to be able to put its progeny or its babies out there in the environment. And so that seems to me like a little bit like Russian roulette, because you're dependent on all these other hosts being where they're supposed to be, acting like they're supposed to act, and so that's taking a big chance on putting your progeny out there. Other questions that I have: how did it know? How did this trematode worm know how to put out an egg that is attractive to the snail so that the snail would take it up? That's a big question, because snails, I'm sure like any other organisms, are pretty particular in what they eat, what they take up, what they allow themselves to be around. And so how did this trematode know to be attractive to the snail? As a cercaria, as that swimming version, once it leaves the snail and now it's in the water column where the fish are, how did it know where in the water column to swim, where to go? Because these things infect specific fish. Not all fish are infected by these cercaria, by these trematodes. They've even done experiments where they've tried to cross-infect, and they don't infect. They only infect specific fish. So how did it know where in the water column to go? You enter the wrong fish's mouth in the water column, and boom, your life cycle is over. You're dead. So you have to enter the right fish's mouth. And I think there's a lot of chance involved with that. That's probably why they put out so many eggs. But what if you enter the wrong snail on the mud flat floor? Boom, your life cycle's over. Game over, you're done. If your snail is not there, if the snail that you infect, that you need to infect, is not there in the estuary where you lay your eggs, boom, your life cycle's over, you're done. If your fish is not there, boom, your life cycle is over. So the snail has to be there, the fish has to be there, the right kind of bird or fish-eating vertebrates has to be there in order for your life cycle to be realized. And so your fish has to be there, and your fish has to stay healthy. What if your fish dies? The fish that you enter, boom, your life cycle's over, you're done. There's a lot of variables that I see here. What if your fish gets eaten by another fish? Boom, your life cycle's over. That fish has to be eaten by a warm-blooded vertebrate or some organism that can bring these cysts to temperature and allow them to exist, to come out of those cysts. If that condition is not satisfied, your life cycle is over. These infect specific organs in the fish. Okay, now you need to understand this. They, Ascocotyle pachycystis will always be found in the heart. Others will always only be found in the liver or the brain or other specific organs in the fish. How does it know when it's tumbling through the blood system? Because what it does when it enters the mouth current, it attaches to the gill, it penetrates the gill, it goes into the blood artery and tumbles through the arteries in the blood system in the fish. How does it know that it's in the right organ? How does it realize, okay, I'm in the stomach now, or I'm in the heart now, or I'm in the liver now, and I can stop and set up shop and make my cyst? If you enter the wrong organ, boom, your life cycle is over. So what's up with the hydrochloric acid resistant cyst? I mean, this is amazing to think about how this cyst is manufactured around the trematode when it's in the fish, whether it's the gill or the heart or the liver, and it makes its cyst, but that whole fish now has to be consumed by an endothermic or a warm vertebrate. How did it know that it was going to have to pass through HCl, hydrochloric acid, in the stomach of the bird or the raccoon or whatever it is that's eating this fish? Where did that come from? That is very, very complex biochemistry. And so to assume that these organisms are sentient beings, that they can think, they can plan, and the literature is full of this language. These parasites have a strategy. They have an end game. They have advantages that they go after. They choose certain advantages. Come on, guys. These are not sentient organisms. They can't think for themselves. And so I see a lot of design in this. What about the fact that these are essentially immune system stealth organisms? Wow. How can they enter the snail and the fish and the bird and not even evoke a response from the immune system? This is staggering biochemistry. They can't know this. They would never know this as a sentient being. And so I see tons of design in this life cycle. Again, I think it's kind of Russian roulette for any organism that wants to mature, become sexually mature, produce progeny, put them out in the environment, and capitalize on the environment to tie itself to this complex life cycle. So I see a ton of design here, but you know, there's a lot to learn, there's a lot to know. So I hope you might consider a career in parasitology. Jump on in and help us try to figure out this stuff. Anyway, thanks. I hope you enjoyed the episode. Like us, give us a thumbs up, send us emails, things you'd like to see on the channel, and thanks again for watching.